Hi, Michelle Glass here. Welcome back to another episode in Chapter 9. We are continuing our discussion of non-Mendelian's inheritance patterns. And today's video is about the pattern of inheritance described as codominance. When you have codominance, you have shared dominance. So in this case, we're going to see that the gene will have at least 3+ alleles in the population. You still will see only 2 alleles inherited for each individual, but there's multiple versions of those alleles in the population, more than just a dominant and a recessive. You're going to see that you would have at least 2 dominant alleles and 1 recessive allele in the population. And so you're going to get at least 3+, really 4+ different phenotypes showing up. So an example in humans is the ABO blood type, where we're going to use the letter I to represent our blood type, and then hang off that with the superscript our different types of dominant alleles. So we're going to have our A allele, our IA allele is dominant, and our IB allele is also dominant, and then we will have a recessive lowercase I allele. Now we're using an I because this characteristic is describing these immunoglobulin markers that are hanging off of red blood cells. And so we can use the I because it's an immunoglobulin marker. Okay, so you've heard of type A, type B, type AB, and type O blood before. Now, in order to get type A blood, you can be homozygous dominant for A, or you can be heterozygous for A. Type B is the same, except it's homozygous dominant for B or heterozygous for B. Notice that type AB is going to be heterozygous for heterozygous for the dominant A allele and the dominant B allele. We're getting equal expression of these alleles, so it is not intermediate. It is not the incomplete dominance pattern. And then we have our recessive, our homozygous recessive form is our type O. Now, what do these genes actually code for? What is this immunoglobulin marker? These are basically the little tags on the cell membranes of our red blood cells that the body can use to recognize what cells belong to you and what cells don't. So I like to sort of simplify things and imagine, if I have type A blood, that I have little A's hanging off of my red blood cell. Of course, that's cartoon drawing, right? That's not really what it would look like. In addition to having these A markers hanging off of your red blood cells, you also would inherit an anti-B antibody. So that means that A would be recognized as self, and any B marked cells would be identified as foreign invading cells, and attacked immune response would be initiated. The same is true for type B, except the B type cell is the cell that's identified as self or normal, and anything with an A marker is identified as foreign, and you'll have an immune response against it. If you're type AB, then you're going to recognize both A and B as normal, and so you don't have any antibodies against these markers because you're going to recognize both A and B equally as belonging to self. If you're type O, you're O because you don't have any markers hanging off of that red blood cell. And so you're going to actually be recognizing type A and B cells as being foreign and initiating an immune response. Because type O cells don't have any markers hanging off them, any person can receive type O blood during blood donation. So we can talk about type O blood as being the universal donor. And then because type AB blood doesn't have any antibodies, it's recognizing A and B equally as self, it's going to be described as the universal recipient. So individuals with type AB blood can accept any type of blood. Okay, that's it for now.